morning, and thank you, Nina. This is almost making me blush. And it reminds me of... Does that work? This is better? Okay. How long I've been in the field, so I don't know if that's a good thing or a bad thing, but thank you anyways for the invitation to speak here about our work. I will be talking, refine my title a little bit about the role of pre-existing memory B cells in human vaccine responses to novel antigens, and with that I mean to antigens that the host has not seen, so in naive individuals. So my lab has been interested in understanding, as Nina was mentioning, the development of B-cell memory for many, many years. And of course, if we think about the textbook concept, we have a naive B-cell repertoire in naive individuals that have never seen a pathogen or a vaccine before. And the idea is that individual cells will bind and undergo affinity maturation in germinal centers and then differentiate into memory B cells, and so this is really the compartment that is important in a vaccine response that is induced, and also antibody-secreting plasma cells. And then upon the boost, the memory B cells can get reactivated or activated and differentiate into antibody-secreting cells as well. And so what we try to understand is how all of this differentiation links to the specificity and binding strength of the antibodies that the cells have on their surface as part of the B-cell antigen receptor, or that they secrete into then the serum. And so we are interested in cell populations, the cell differentiation paths, and then the individual molecule. And we work a lot with structural biologists and then also computational people. And then we want to use this knowledge to help instruct vaccine design. And so for many, many years, my lab has been working on trying to understand the response to Plasmodium falciparum. This is a unicellular parasite that is injected into the human skin by infected mosquitoes. And from the skin, the parasite that has this kind of banana shape here goes very quickly from the skin into the liver. That takes about an hour, and then infects hepatocytes, differentiates and multiplies a lot, and then comes out as something that looks completely different, also has completely different gene expression profiles, and then infects red blood cells, and that's what's causing disease. And so we are interested in the response to the protein that is densely coating the surface of the so-called sporozoite stage, the stage that is injected into the skin here, which is a protein that is essential for the development of the parasite in the mosquito vector and also in the mammalian host. The protein has a very conserved region that is the major B-cell epitope, so it's great as a vaccine target, and it is actually the target of the only available malaria vaccines. And many, many have been tried, and only these two are available. They are actually very similar, and they both target this antigen on the surface of the parasite. But what is also true is that while it's great that we have these vaccines, their efficacy is really low. It requires 4 immunizations to get to a maximum of 50% efficacy, and it's not lasting, so it's only about 1 to 2 year durability. So when we started working on this problem, we wanted to learn more about this and see whether we could improve the vaccine, and I won't be talking about this today, but just some findings or some observations that we made when we looked at immune responses in individuals that were naive to malaria because they live in Europe, and that were exposed to live parasites under chemoprophylaxis. And so this is considered an immunization strategy that you inject a large number, 50,000 of these parasites into these people, 3 immunizations, 4 weeks apart, while you give this drug, and that's the same drug that you take before you go to Africa when you are might be exposed to Plasmodium falciparum. And so this drug prevents parasite development when the parasite comes out of the liver and kills it. But the parasite can still develop in the liver, and the immune system can react to the injected sporozoites. And so we studied the response always 1 week after each vaccination in these individuals. We could get blood samples, and so we used the fluorescently labeled CSP protein, this surface protein of the parasite, to isolate memory B cells that were generated over the course of these immunizations. And what you see, because these, as I said, were naive individuals, they had never seen the parasite, we see no memory response before the immunization, no labeled cells, but with the immunizations, the frequency starts to increase, and every individual develops a memory B cell response against this target. Then we went ahead because we wanted to understand the quality of the antibodies that these cells make, and generated monoclonal antibodies from these isolated single cells. And then now here every circle is the reactivity, is showing you the reactivity of a monoclonal antibody from one of these cells. And this is here the major B cell epitope of the CSP protein. And you see that initially the quality after the first immunization of the antibodies that are made by the cells is still low, but with repeated immunizations, we get a very strong and good binding response here. And because these antibodies are all functional, we also see that these antibodies can inhibit the parasite very well in an in vitro assay that reads out blocking the parasite before it can inject hepatocytes. And so, of course, this looks like perfect affinity maturation here. And so when we look at now the protein binding capacity of the antibodies here as read out by ELISA versus the number of mutations in these cells, we see two things. So first we see that actually there are many of these newly generated memory B cells have no mutations, suggesting that they were really developing from naive precursors. And some of these had very high binding capacity without any mutations, suggesting that there's a really good number of potent precursors in the human repertoire that can bind to this protein and then also inhibit the parasite quite well. And so you see how the frequency of these cells increases over time, over the immunizations, and then you also start to see that some of these cells seem to mutate and gain mutations and hopefully also, and we've shown that, affinity to the target protein. But what we also observed, and this was actually initially quite puzzling, is that after just 1 week after the first immunization, we see that many of the binding memory B cells carry actually very high numbers of mutations. So these mutations could not have been accumulated within the 1 week time frame after exposure to the parasite. And so they must have been pre-existing, and you see that these pre-existing memory B cells with these highly mutated IGs that they express were always present after all 3 immunizations. So they also bound the protein, but they did not seem to do a lot here. And so the question for us was, when the response, initially these cells get recruited into the response, as I'm showing here, in these naive individuals after just 1 immunization, what happens to them? Do they also undergo affinity maturation and gain higher affinity to this new target because they are initially a little bit cross-reactive and then they change their focus basically, or not? And would they do something similar to the naive B cells that we observed? And so we learned a lot of other things from these studies that I don't want to go into now. It's all published, and so I just highlight the papers here. So the question for us was then, is the recruitment of memory B cells unique to malaria and this CSP response, that these pre-existing B cells get recruited into responses in naive individuals? And how do the naive and memory B cells behave in such a naive response? And what is the quality of the IGs or antibodies that they express? And so when SARS-CoV-2 hit us, there was a unique opportunity to actually study that in real life, in people. And so what we did, we took blood from individuals before and then over the course of 2 immunization with Comirnaty, so the mRNA BioNTech vaccine, Pfizer-BioNTech vaccine, and we sampled always 1 week after the first dose for 3 weeks, and then there was a time between. The boost was given after 6 weeks, and then we sampled again at the time of the boost and the following 3 weeks. And so these individuals mounted a very good serum response against the vaccine. These curves here are the vaccinees, and you see that after, this is the IgG response, after the prime, titers go up as expected, and then after the boost, they shoot up more and they stay very high, and they reach the level of an individual that was actually previously infected with SARS-CoV-2, and so in this individual, the vaccine induced very strong responses to start with. There was no IgM response really, and also no IgA response. I'm not showing this here. And so when we look at the cellular level using flow cytometry, we saw that there was an increase in antibody-secreting cells after the prime, which was expected, and then there was not much of an increase after the boost. But at the same time, we see that the frequency of spike-reactive memory B cells, so the vaccine target, increases over time here, and also the frequency of spike-reactive naive B cells. And this goes pretty much in parallel. After the prime, it drops a little bit, and then with the boost, there's an increase again. And here again, we see that the majority of spike-reactive cells are actually not the naive B cells, it's actually the memory B cells that bind this frequently. And so we assumed that when we bring this together, the high titers here is not really just linked to higher numbers of antibody-secreting cells, but probably also to higher quality antibodies. So see that, and that's all expected. So the question here then is, what happens to these naive B cells and memory B cells in these individuals? And what we see here is likely that the naive B cells probably also differentiate into memory cells and then increase this frequency over time. So we then wanted to understand more about all of these cells, and we isolated then cells using barcoded spike protein to get to transcriptome and repertoire data using 10x. And we isolated 3 populations. So we sorted spike-binding B cells, naive B cells, and also pre-existing memory B cells, or memory B cells after the vaccination. We sorted antibody-secreting cells using pretty much CD38 as a marker without, independent of their spike reactivity. And we also sorted activated cells using CD71 as a marker. And again, we looked at naive and memory B cells. And so when we plot here the CD71 positive, the frequency of CD71 positive cells among the spike reactive cells, we see that there is initially not much happening, but then with the boost especially, the frequency goes up of these activated cells in the memory, and also here in red in the naive compartment. Whereas very little change is observed in CD71 positive cells that did not bind the spike protein over time. There was quite a number of, or a frequency of activated as negative memory B cells throughout here, but there was little change, so we ignored these cells from then on because we thought they reflect bystander activated cells, and we focused our analysis on these S positive cells. We then looked at the transcripts of these cells, the profile. We could distinguish these cells as expected, similar to the flow. We identified naive B cells, memory B cells, and antibody-secreting cells, and this is just showing you how these cells, distributed over the different time points. And then we looked at the profile. We see that memory B cells still looked more like naive B cells, whereas antibody-secreting cells were quite different. But these memory cells were really memory cells because they carried mutated Ig genes, and they were almost all class switched, and very similar here to the antibody-secreting cells, whereas of course the naive cells were not mutated and not class switched. And the naive cells were also not clonally expanded, in contrast to the memory cells and the antibody-secreting cells. And to show that these naive cells participated in the response, we looked also at clonal expansions prior to the immunization among these cell populations. We see that some of the clones persist over time, so that's here. And so they really probably participated in the response. The question then was, do we see the same using the bar-coded spike, what we've seen with the flow assessment, and here we see again antibody-secreting cells go up after the prime. There's a little bit more of a peak that is detectable after the boost, but still the large numbers of antibody-secreting cells are seen here, and these are just S-reactive cells, so they bind the spike still, so it's also likely that we detect fewer of these spike-reactive antibody-secreting cells because they downregulate the BCL receptor as they mature to become terminally differentiated plasma cells. And then we look at spike-reactive naive B cells and memory B cells, and here you see that memory B cells are there, and then they come up after the boost, and the naive B cells are also there. Their frequency decreases after the boost, again, probably because these naive B cells differentiate into memory B cells. And so when we then look again at CD71, we again see that the spike negative cells don't really change over time in their frequency, and so we ignored these, as I said, and think that the rest is, these cells are bystander activated, and we really focused only on the S-specific vaccine response. So then we started looking at the Ig genes of these cells, and we're now focusing on spike-reactive IgG memory B cells and look at their somatic hypermutation pattern, and we see that a lot of, similar to the malaria, a lot of the pre-existing memory B cells that bind spike have already high numbers of mutations, and then after the first immunization, this is really the dominant population here. And then with time and over the boost, we see that these unmutated or lowly mutated cells start to come up as a population and increase in frequency. And there is really this bimodal distribution of the somatic hypermutation load. And so we marked these here, setting a threshold as a low somatic hypermutation population in contrast to the ones that carried high numbers of somatic mutations and were very similar to the pre-existing and the early cells. So from now on, I will refer to these low and high SHM populations, and you see that the frequency of these low SHM cells increases over time, likely because these are activated naive cells that start to accumulate mutations, whereas the frequency of these highly mutated cells did not really change much over the course of the immunization. And so this is different from IgM memory B cells that also bind the spike. Here we see the same two populations pretty much, but there is no change in their frequency over time, suggesting that they didn't really participate in the response. And so we didn't really look much deeper into this. So then we decided to look again at the frequency of these high and low SHM cells and spike-reactive cells. And so here we're looking at the frequency of S-positive IgG memory cells, and you see that the cells with low mutation frequency increase here and dominate the memory B cell response with time, whereas the spike-positive, highly mutated cells decrease in their frequency over time relative to these. So the response is really driven by the naive compartment, but these cells that were pre-existing are still there. And like I said, there is not really much in the IgM compartment. When we look more closely at these two high and low SHM populations in the IgG compartment, we see clonal expansions, and we see them more in the population with the high mutations than in the ones with the low mutations, and so obviously this change is reflected by the diversity of the populations, clonal diversity. And we also see that overall, the highly mutated IgG memory cells have more clonal expansions that have longer branches here, and so seem to have been accumulating mutations for longer, whereas these are the newly formed low SHM kind of trees that we see, very small clonal expansion yet. When we go into the transcription profile, we also see that these two populations have different transcription profiles. So here, black is the low SHM and white is the high SHM population. And so we look at these clusters here, and we see that this separates nicely according to the mutation patterns. And so we see that there are unique clusters of transcripts that are associated with both of these populations, and that they are very different. When we look more deeply into what are these genes that are expressed, we see that it's, there's, I just summarized this here. The low SHM population looks like these cells have been coming out of recent germinal centers, whereas the high SHM population looked like it was more going into the plasma cell differentiation pathway. There were a lot of transcripts associated with cell motility, T cell help, activation, and so on here, and more of the plasma cell transcript signatures in the high SHM population. So it seemed that the naive B cells that got recruited into the response underwent affinity maturation, clonal expansion, and differentiated into these S-reactive memory B cells that we then read out here, whereas the pre-existing memory B cells that also bound the spike protein seem to differentiate into antibody-secreting cells. And so this was surprising a little bit because these people had never seen SARS-CoV-2, and so this didn't look like the original antigenic sin idea where we have high binding pre-existing memory B cells and they would just take over the response as reported in flu. So the question was, are there differences in the S-reactivity between these naive B cell derived memory B cells and memory B cells that came from cells that had already been mutated and pre-existing? So we looked initially at barcode level, and we could see that the low SHM population bound the spike protein overall better than the high SHM population, and the same was true when we zoomed into antibodies or cells that bound the RBD part of the spike, so the ones that's really important to bind the host receptor. And we could also see that these two populations differed in their gene usage, Ig gene usage, and so seemed really different. And when we look more deeply into exactly which VH and light chain genes they use, and focus on genes that are associated with spike reactivity based on what has been published in the literature, we see that the low somatically mutated cell population has more of these, uses these genes more frequently compared to the high SHMs, including here the JH6 and VH353, 366, which has been associated with high affinity to the spike protein, which is really enriched in the low SHM population, and also shows overall longer CDR3s and another signature. So low SHM seem to bind the spike protein better and so have higher binding antibodies rather than the high SHM pre-existing memory cells. We then looked at antibody, at recombinant monoclonal antibodies that we made from these cells, and we made a total of 166 antibodies. We had some control. This is a positive control antibody here. We see that 93% of the cells that we make monoclonal antibodies from really then also bind in this ELISA assay, and there's a few that don't, which is a technical background sort of. We can look at all antibodies from all the donors and look at the high-end low SHM-derived monoclonals, and we see that in each donor, antibodies from this compartment bound the spike protein better than the highly mutated. And when we look at all the data together and we separate it by time, when the cells were isolated, we see that the highly mutated memory B cells make antibodies that don't really change in their affinity over the immunizations compared to before the immunization, whereas the ones that are derived from low SHM, so naive B cells, do. And this binding strength is clearly different from naive B cells that were isolated before the vaccination. And so when we then look at the strong binding B cells, we also see that the frequency of strong binders that are here in this ELISA with this high AUC is much, much higher in the low SHM memory B cells than in the high SHM memory B cells. We then look more deeply into what exactly do these cells bind in the spike protein. We can separate the binding to the RBD, receptor binding domain, or here the S1 and S2 part of the protein. And so we see that many of the cells bind either the RBD, which is part of the S1 domain, so most of these cells bind this part strongly, and some cells bind, their antibodies bind the S2 domain here. And we can again look at this RBD binding then also in ELISAs and see again that binding to the RBD is also stronger in lowly mutated memory B cells. And we can also confirm this by surface plasmon residence affinity measurements as shown here. And for this population, then the affinity to the RBD correlates with the somatic hypermutation load, again suggesting that memory B cells that have still low somatic hypermutation level originated from naive cells that underwent affinity maturation, accumulated mutations, and were selected for higher binding in germinal centers. The question for us was, these pre-existing memory B cells that bound spike didn't really change in their affinity over time. Could they have been recruited into the response because there was cross-binding to common human cold viruses, and this has been reported in the literature, and we all constantly exposed to these viruses. So we tested all of the antibodies for binding to a number of cold viruses, and we find some antibodies that actually do bind some of these viruses, not all. And these are all antibodies that bind to the S2 of SARS-CoV-2, and that is because this is the conserved region of the spike protein. So that all makes sense. And then we have a fraction of cells that just bind everything and seem to be unspecific. So these cross-binding to these common cold viruses was also seen basically at serum level. There was a titer in all of these individuals for these common cold viruses, but the titers didn't change over the vaccination, suggesting that this was really nothing that had anything to do with the SARS-CoV-2 vaccine response, or at least not much, and was really just cross-binding, but nothing specific. So we tested then these antibodies that were cross-reactive and everyone else here for polyreactivity in a very sensitive ELISA to detect whether they bind to anything here. And we use DNA, insulin, and LPS, and we see, yes, there is a fraction of antibodies that is unspecifically binding to these antigens, but the frequency is actually very low, and this is independent of whether the cells had high or low somatic mutations. It's the gray part here in this graph. So we see that there is a difference a little bit between these two populations in that the high SHM pre-existing memory B cells are more frequently here cross-reactive with the HCOFs, which is expected. But as you see, the reactivity is not very strong. And then we see more of the RBD and spike really binders against SARS-CoV-2 in the naive B cell derived low SHM population. We can look at this over time, and there's nothing really major to see except that this reflects at both time points after prime and boost, we see the same thing. So again, what do we see? There is some low level of HCOF cross-reactivity that is focused on the conserved S2 domain in pre-existing memory B cells more than in naive B cells. So it exists in both populations, but it's more here. And then still we see that these pre-existing memory B cells, although they do not bind the spike protein well of SARS-CoV-2, they just seem to differentiate into plasma cells. Whereas the naive B cells clearly affinity mature and then differentiate into memory cells. So the question then was, do we really see that these cells differentiated into antibody-secreting cells over time? Although they don't show signs of affinity maturation and no higher affinity, which was previously linked to earlier differentiation of memory cells into blasts. So cells with higher affinity are always believed to immediately differentiate into blasts and not re-enter germinal centers, whereas low-binding cells should enter the germinal center and then mature and then differentiate into antibody-secreting cells. So that's what we wanted to understand. What about naive B cells? Do they differentiate into plasma cells or antibody-secreting cells as well, compared to these cells? And so I apologize that some of these images are blurred. I don't know what happened. I tried to fix it, but it couldn't. So we looked now at the antibody-secreting cells that we had isolated and focused on the ones with the isotype switched, and we see the same two populations come up. Low SHM cells can be differentiated from high SHM antibody-secreting cells, very similar to what we've seen in the memory compartment. And so we see here the same trend, so low SHM antibody-secreting cells accumulate with time, whereas the ones with high SHM decrease over time. And this is, again, very similar in the IgM compartment, antibody-secreting cells. We see the two populations, but they do not change over time, and I'm not showing this here. Very similar to the memory B cell compartment. So when we then look at the gene usage of, this is heavy and light chain, for the antibody-secreting cells that are here in lighter blue and red, and apologies again, and we separate the high SHM population in blue from the low SHM population, we see that the two populations clusters together. So low and high, low SHM memory cells and antibody-secreting cells cluster together, and high SHM memory and antibody-secreting cells cluster together, suggesting that they are linked and clonally related, probably. And indeed, we see some clonal overlap here. So low SHM antibody-secreting cells have high clonal overlap with low SHM memory B cells, and the same is seen for the high SHM population. But there is no clonal overlap between these low and high populations. And so I guess, this is wrongly animated, but yeah, this antibody-secreting cell is very similar to the behavior of the memory cells. We then cloned some antibodies from these antibody-secreting cells, just same as we did before, not as many. We found the S-spike reactive cells, and we see, again, the same thing. Antibodies from high SHM antibody-secreting cells had overall low reactivity to the spike protein, whereas the ones from the low SHM really had higher binding strength, and we can summarize this here. And specifically, also the strong binders were more frequent in the low SHM compartment. And again, I'm showing you again for comparison the IgG memory B cell compartment. It looks very, very similar. These low SHM antibody-secreting cells also had on average more spike reactivity with the bar-coded spike protein, as you see here, again similar to the memory cells, and also the frequency of S-reactive cells was higher. So we then try to understand the differentiation path of these cell populations, low SHM and high SHM antibody-secreting cells. And Anna Obratsova, a student in the lab, performed this pseudotime analysis. And what she could show is that over the time, the cells lost basically proliferation markers, strong B cell markers, and pro-apoptotic markers, and instead plasma cell markers, and these pro-survival markers went up. And so she could link this to the cell differentiation from plasmablasts, so early activated antibody-secreting cells that are still proliferating, and then sort of more early plasma cells and really mature plasma cells that then start to express really CD138 and so on. And so when we look now at cells that have low somatic mutations or high somatic mutations, we see that the low somatic mutation population is enriched among these early antibody-secreting cells, whereas the high SHM population really is very abundant in the more differentiated mature plasma cell compartment here. And the low SHM, they get there, but it takes them time to get there over the vaccinations, as you see here. So this brings me to the summary of the findings. So in the SARS-CoV-2 naive individuals, what we see is that mRNA, the vaccination recruits naive and also pre-existing memory B cells into the response. So the repertoire is not just this, it's all of the cells, and in humans you have about 50% naive B cells in circulation and 50% memory cells, about half may be class switched. And so there's the system draws from this pool of cells and recruits these cells into the response. But it recruits the naive B cells into germinal centers where they undergo affinity maturation and then become memory cells, whereas these pre-existing S-reactive cells don't seem to change their receptor anymore. They don't seem to go into germinal centers, although, of course, we haven't sampled germinal centers, and others have seen them in germinal centers. We think it's probably still possible, but maybe there's a threshold that the VCR needs to show to get into it. The vast majority of these cells seems to just become antibody-secreting cells, although their receptors don't bind better than the ones from naive B cells. So despite this low S-reactivity, as I said, these cells dominate the early antibody-secreting cell response. And so the really high affinity cells that come up over the immunizations and that are induced by the vaccine response really all come from these naive B cell precursors, as probably expecting, and not from these cells. And they accumulate in the memory compartment and also with time in the plasma cell compartment. So with this, I want to acknowledge the people I mentioned, Anna Obrazova already. She did all the computational work. Lee did all the experimental work, a postdoc in the lab, with support from the people that are highlighted here in bold, our collaborators in the institute, the funders, and then lastly, I'd like to mention to anybody who's interested in coming to a B-cell meeting, we are organizing one in Monaco next year in February, so maybe a nice place to visit. Thank you.
Can you hear me now? There we go. Okay. So let me just start by asking a question, and then we have a lot of nice questions in the Slido. Do you think that these high SHM cells that you see that potentially are poised to go to antibody-secreting cells are similar to the types of cells you're seeing in the malaria response? Is there a connection there that you might want to comment on?
Yeah, I don't think that we can define them as a specific population based on their phenotype. Is really probably just their capacity to bind to the antigen at low level. So this kind of cross-reactivity is probably expected, right? Antibodies never have just this one antigen that they can bind to. They maybe bind one with high affinity, but then it's a curve, right? So, and there's some cross-reactivity always. It's just the question when it is high enough to activate the cell.
Yeah, or maybe they're not fully functional in a good way, right? That in the response to malaria you have issues.
That's a different population. Malaria, we know there's this atypical memory B cell population, but that's yet another population.
Okay.
They can make very good antibodies. It's just that these cells seem to be exhausted because they're overstimulated in a way.
Great. Okay,
That's
so
a different population.
Okay, so we're going to get to some of that in the Slido questions too, which you can see up there. So the first question is, how did you set the threshold between low and high SHM cells?
Yeah, that's what really looking at the distribution of the somatic mutations, and it was pretty bimodal. So we have that the data is on bioarchives, and hopefully it's soon going to be published. So you can look into that, and it was a threshold that we set.
Okay. Second question is, did you try tracking clones of the high SHM cells over time? Are they the same clones that persist over time?
Yeah, so I think that's always a question of sampling depth, right? We didn't really sample so deeply, but yes, we see clones that we can track over time. And as I showed some, right, some have these branches that are longer, so it's easy, and they are larger, so it's easier to track them over time, but yes.
But do you think as a population, just trying to feed into the mind of this person who asked it, do you think as a population that most of the clones that are in that high SHM group are long clones, or are some of them actually crossing over from other populations?
We can never
Never
totally
really know,
make,
right?
no, but we know that some of the pre-existing clones were tracked over time. So yes, they must be persistent. They can be persistent.
They certainly can be, yes. Okay, great. So how confident are you that this is truly a naive response? I'm guessing here they're referring to the low SHM cells. Could the subjects have been primed by exposure to related coronaviruses?
Yeah, this is what we tested with these human, right, the cross-reactivity to the common cold viruses. So there was some, but it was this, and a little more in the high SHM population than in the low, but we didn't detect any high binders. And yeah, and also at serum level, there wasn't a high response.
Yeah, yeah. Have you tried using computational methods to improve the spike affinity of the high SHM cells? That just
No.
in there?
No, I'd love to do that.
If the spike negative cells were activated by bystander activation, wouldn't their levels correlate with the spike positive cells?
So the bystander activation, we think we only see for the cells that express CD71 that we isolated, not for the others. Because then again, we can see the clonal overlap with the antibody secreting cell population with high somatic mutation. So that shows that they can really participate in the response.
So do they correlate between the CD71 positive spike positive and the positive spike negative? Not really.
No, we did not maybe look deeply into that. I would have to ask Anna to look into that. I can't answer.
Okay. So have you observed / analyzed the response of other B cell populations? This comes up now again. Yeah, i.e. atypical B cells.
Yeah, atypical B cells, we looked at those cells in the context of the malaria. I am not a fan of the term atypical, to be honest. But it's a population that is detectable in malaria. It's also detectable in people with autoimmunity. And in immunodeficient people, I think it's an activation phenotype that is sort of this overactivation. And many of the cells in malaria made perfect antibodies to the parasite. So it's not that they're not functional. And we detected also secretory Ig transcripts in those cells in malaria. So again, saying that they probably did secrete at some point. But when we fish them out with this phenotype of CD21 low and everything, then they're probably not that functional anymore.
Yeah, and did you want to comment any further about atypical B cells and the flawed nomenclature?
Yeah, I think atypical has a meaning, right? I prefer to describe them by their marker expression rather than by some function that no one has shown and that may be different in different diseases also, right? It's again, if you're immunodeficient, you also have hyperactivation of the few cells that are in the system and that need to make up for the lack of the other cells. So I think there can be many reasons to get to the same thing, but yeah.
Yeah, so many pathways to getting to that phenotype, and you're not really sure what you're looking at.
Exactly, I fully agree.
Yeah. Okay, so do you observe new naive derived cells after the vaccine boost? And so how were they differing from the B cells induced by the first dose?
After the boost, we also see unmutated cells that get recruited into the response. So yes, I would say, you again bring in naive B cells, and I should say that not what we describe here in this system, there is a mouse data from Gabriel Victora's lab and others that have shown that the germinal centers are open, right? Naive B cells can get recruited, and with the booster, you also probably make new germinal centers. So yes, naive B cells probably go in. And actually, interestingly, in the mouse, the data looks similar, that the memory cells don't really seem to re-enter the germinal centers. So that's not different from maybe here, although we don't sample germinal centers. So.
Well, so does that kind of counteract original antigenic sin then, in a way?
So again, I think it's puzzling, right? Because the original antigenic sin, clearly then the cells take over and suppress the response of the naive population. I think it's all a question of affinity, and as I said, the data seem to suggest that there is some kind of affinity threshold maybe for memory cells to re-enter, maybe? Because clearly for flu and original antigenic sin, if you have very high binders, that has been shown to happen.
Yeah, if you go back to the bottom, we haven't quite finished.
And I think there is new data coming from other labs probably around the same question in the context of diseases and other vaccines. So.
Yeah, no, it's a really interesting, it's very interesting. Another question here is, can you speculate on the reason why the low SHM have a higher affinity? Could it be to counteract effector function?
So they don't have a higher affinity when they enter the response. It seems very similar, although of course affinities we can't really measure at that level. It's really just based on avidity measurements using ELISA. I think that's important to keep in mind. They don't seem to be different. I think what is a naive cell, of course, has an IgM, and it's different in signaling compared to an IgG on a memory cell and all that. So. I think it must be the composition of the cell, right? Signaling capacity and the BCR signal versus other signals that are integrated at the cell level to tell the cell what to do. And so that drives the low SHM naive cells into germinal centers and the others probably not. And it's known that memory cells are more prone to differentiate into antibody-secreting cells. That's old literature, with TLR stimulation and non-BCR signals can drive them easily.
And atypical, naive, apropos of Inaki-san's, right? Yeah.
Yeah.
That was a very popular theory during COVID, right?
Yes.
Had these atypical naive that would become activated.
I think a lot of things are possible. It's just whether we can see all of this in our study. I think we have to be careful here. I don't know about counteracting effector function and so on.
Great. Okay, so we'll double check back in with the Slido in a minute, but are there any other questions from the floor? I think I see one.
Really nice talk. I was curious if you've mapped the specificity of any of the high HSM cells that are recruited early on.
You mean the epitope mapping, really, of those cells?
Yeah, identifying their antigenic specificity. Where they
the high SHM cells, whether they have some other, their real target, you mean, to identify that?
Yes.
It's a super interesting question. We haven't done that. I'm very interested doing that actually also with the malaria cells. I assume it's some microbial target, right? Gut microbiota comes to mind. Actually, it's interesting because not so much in COVID, but in the malaria response, we see a lot of IgA cells, memory cells that initially get recruited into the response as well. So IgA cells also there in COVID, and I'm not showing it here. But
IgA
memory cells, so pre-existing, S-reactive IgA cells, but in COVID it's difficult because that could be, again, more of those common cold virus related cells. But in malaria, I think it's a cleaner readout.
Thank you.
Any other comments? I see one more question on the Slido here. So when you screened against human coronaviruses, did you look at old variants?
So no, I can't say that we used the 4, most common viruses that we could think of, and no, we haven't. Of course, you can never exclude that there's something that, these specific hosts have seen, but I think it's, the pattern is the same in every donor, and I understand the limitation of 4 donors. So I think these findings need to be confirmed in other systems, and don't have an answer.
Okay, I think so. Hedda, for her really great talk.
Thank you very much.